Hello everyone, I'm Tejal Salunke. I'm an intern volunteer at Forensic Fusion. Today we'll talk about what is drowning and what are diatoms. Before we move into the main topic, let's know about what is biology. Biology is a study of life and living organisms, including their structure, taxonomy, etc. It is the foundation for understanding the complex processes that sustain life. Then what is forensic biology? Forensic biology is a specific application of biology to legal matters. It involves the analysis of DNA, blood, and other bodily fluids for investigations and legal proceedings. So let's today learn about what are diatoms. So in this we'll be learning about what is drowning, its types, what are diatoms, its types, tests for diatoms, then extraction methods and microscopic techniques. We'll also learn about the medico-legal importance of these, its consequences and advancements, and some case studies. So moving forward, first let's start with what is drowning. Drowning is a form of violent asphyxial death caused by aspiration of fluid into air passages caused by complete or partial submersion in water or other fluids. So it basically tells that the death occurs either due to the entry of fluid in the respiratory passage or due to the effect of severe water and electrolyte imbalance. So when a person enters or has been enters a water body, a lot of water enters a person's body, like through nose or mouth, and then this water it blocks the air passage basically, which leads to death. Or excess water content in a person's body or different electrolytic imbalance that can cause leads to the person's death. Then talking about types, there is wet drowning, dry drowning, secondary drowning, and hydrofusion. So what is wet drowning? This is like the most common type of drowning in which the water is completely inhaled into the lungs. So in wet drowning, the water enters the lungs of this person. Next type is dry drowning. In this, what happens is the water enters the larynx. So we have a part here called as larynx. So when water enters, it accumulates near the larynx and the person is not able to talk, which leads to the person's death. Then talking about the secondary drowning, it is also called as post-immersion syndrome. In this, so it's not mainly due to water or because of any causing the breathing to stop or anything. What happens in this case is when a person is thrown into a water body or he falls into a water body, there can be external traumas like head traumas or anything. So it's mainly due to head traumas. When a person falls, if he or she hits a rock, then there will be a head trauma which leads to the death. And talking about hydrofusion, this is also known as immersion syndrome. Okay, so what happens in this is when a person at normal temperature enters a very cold temperature, that is very cold water, comes in contact with very cold water, he or she gets sudden shock which leads to the death, basically causing cardiac arrest, which is the cause of death. Next, moving on to diatoms. Diatoms are photosynthesizing algae that have a siliceous skeleton and are found in almost every aquatic environment, including fresh and marine waters. They are even found in soil and everywhere wherever there is moist or where there is moisture basically. So diatoms are like, these are infiltrate that are found inside the body of a deceased. These usually serve as corroborative evidence or sometimes even conclusive evidence to that help in supporting the diagnosis of the death. These usually help in ascertaining if the drowning was ante-mortem or post-mortem. We'll be coming to this ante-mortem and post-mortem in the further slides. Talking about the discovery of it, it was first discovered in lungs by Hoffman in 1896, and the diagnostic was done by Ravenstoff in 1904. Talking about the types of diatoms, there are mainly two types, that is centrales, the centric diatoms, and pinnales. These are the pinnate diatoms. So the centric diatoms are radially symmetrical with parts radiating out from a central point. And the pinnate diatoms exhibit bilateral symmetry, meaning the left and the right halves are mirror images of each other. Then talking about the diatom test, so the test is usually based on the presence of diatoms in the water where the drowning has likely occurred. It tells if it was a ante-mortem drowning or a post-mortem drowning, as we can see in the image here. So when a person who's alive is put into the water body, basically where we say it can be a murder or he tries to do suicide. So a live body enters the water. What happens is, when a person is alive, the heart beats, which leads to circulation. So because of this process or mechanism taking place inside our body, when a live person enters a water body, the water enters our body through the nose and mouth, and it circulates along with the blood. So it helps the diatoms, or it makes the diatoms to reach inside the lungs, the stomach, and also in the bone marrow. So in case where the person was alive and then was found in a water body drowned, in case of post-mortem, after the post-mortem is done, we'll get to know, like, if the diatoms are found in the person's lungs, stomach, and the bone marrow, then it's understood that the person was alive when he was put into the water. But in case when a dead person is thrown into the water body, the diatoms tend to be found only in the lung area. It do not travel a lot, since there is no circulation happening in our body. So the heart won't beat, there won't be any circulation of blood. No circulation of blood leads to no circulation of these diatoms. So these diatoms will be found only in the upper areas, like just in the nasal cavity and the lungs to the max. It won't travel till the bone marrow or the stomach. Next, moving on to the extraction methods, there are four types: acid digestion, enzymatic digestion, Salunke 350, and microwave digestion. We'll talk about the acid digestion in a while. So before that, what are these enzymatic digestions? So these enzymatic digestion usually involves using proteinase K to mince tissue samples. These are rinsed in the solvent, then centrifuged, and the solid residue is removed and observed under the microscope. If we talk about the Salunke 350, it's a method that effectively extracts freshwater diatoms by washing tissue samples, again centrifuging, suspending the residue in Salunke 350 solution, and incubating overnight, and then observed under the microscope. If we talk about the microwave digestion, it is considered to be as a very sensitive and efficient and less contaminating method for the extraction of diatoms, which is again then observed under SEM, that is scanning electron microscope. Talking about the acid digestion, here there are two types of acid digestion, that can be nitric acid or sulfuric acid. If we talk about the here, let's take an example of nitric acid extraction, where so what we do here is, we'll take the bone marrow from the femur or the sternum, and we'll keep this sample in the beaker with concentrated nitric acid in it. We'll leave the sample undisturbed for 2 to 3 days, and then later on the extraction or the sample has been then taken and centrifuged for 30 minutes at 2000 to 3000 rpm. The supernatant has been separated out, and this is further again added with 20 ml of water and centrifuged again. The centrifugation process is usually carried out for at least 2 more times, and then the final sample has been observed under the microscope, where the different features and the structures of diatoms have been observed. So next we'll talk about the microscopic techniques. These are nothing but so after the extraction process is taken out, we usually observe the samples under microscope to see the proper structures of it, and also if there are any foreign materials found in, we'll just these have been removed out from the tissue samples. So what are the microscopes used? In this we use light microscope, scanning electron microscope, transmission electron microscope, and atomic force microscope. So next, talking about the medico-legal importance of it. The first and foremost thing is like to know if it was an antemortem or a post-mortem drowning. In case where it was the antemortem drowning, we'll find presence of froth, and there will be a cadaveric spasm. This basically means when a person enters a cold region, so a sudden stiffening of the body takes place. So when cadaveric spasm happens, that is sudden stiffening of the body, the person's hands tend to roll, like we tend to take it in a fist form, where we try to grab whatever is around us. So usually in these conditions we find the soil or seaweeds which were present in that area in person's hand. Then we'll find water in middle of ears and in stomach, and it is a positive diatom test. So when we talk about the medico-legal importance, like where there are pros, there should even be consequences of it, like cons. So talking about the consequences, there are 2 consequences. That is, one thing is false positive and another one is false negative. So as we know that diatoms are not just found in water, it's even found in our food, it's found in any place where there is moisture, it's found in soil, everywhere. So what happens is, in false positive result, it sometimes give positive results for presence of diatoms in our stomach or in our bone marrow, which we usually confuse with drowning. Like we think it may be due to the drowning that diatoms are present in our stomach, but the main reason behind it is that diatoms are present even in our food that we consume on a daily basis. So this leads to false positive. Okay. So when we talk about false negative results, so what happens here is, like in the case of hydrofusion, our sudden change in the temperature which leads to cardiac arrest of the person, it suddenly stops the beating of heart and leads to person's death. So in this condition, it's not really possible for diatoms to travel through a long distance and reach our stomach or the bone marrow. In these conditions, the diatoms won't be found in the organs, so it can give false negative results too. Talking about the advancements of it or the future potentials of diatoms, we can say that diatoms are really helpful in solving cold cases. What are cold cases? Cold cases are those cases that did not have any conclusion to it, basically were been closed due to lack of evidence. What significance does diatom play here is that diatoms tend to be on clothes for a longer period of time. Even after like 10 years, 20 years, it's still there and it can be analyzed. Like proper way method, if it is followed, then we can analyze diatoms even from clothes that were been like that acted as an evidence for a drowning case like some 20-30 years ago. Next, let's do a comparative case study. We have like two case studies. In one, there is a 19-year-old boy who has been found submerged in a water tank. There was no sign of injury on this person's body. There was no sign of injury found during the time of autopsy. The nitric acid extraction method was carried out, and it was found that the diatoms found inside the person's body, this boy's body, was same to the diatoms found inside the water tank. So it was later on concluded that the boy died due to drowning. So what really happened here was her husband had an extramarital affair, and so he killed her and then later on dumped her into the canal, and then pretended or tried to show that it was a suicide that she committed, but it was not, and she was been murdered, which was later found by diatom analysis, as the absence of diatoms in the bone marrow showed that the water did not enter the person's body, which means the person has to be dead before entering the water body. There are a few references listed down here, so you can go through these references for more detailed study about the topic. Yeah, that's it. Thank you. I hope this session was useful to you in some way. Thank you so much for joining me on this journey into the world of diatoms and forensic science. If you have any questions or any comments, please feel free to leave them down below in the comment section. Also, don't forget to like, share and subscribe to Forensic Fusion for more such content related to forensic science. Thank you.